This video will cover the formation of imines. An imine is the nitrogen analog of a ketone or aldehyde. An imine is synthesized by adding an amine to a ketone or an aldehyde under acidic conditions. In the first step of the mechanism, the carbonyl oxygen attacks the proton, forming the protonated carbonyl and increasing the electrophilicity of it. This protonated carbonyl has a resonance structure, which helps us to understand why protonation of the carbonyl makes the carbon of the carbonyl more electrophilic. Once the carbonyl has been protonated, the next step is attack of the nucleophile, in this case the amine. This produces the tetrahedral intermediate. Deprotonation of the tetrahedral intermediate by another molecule of amine produces the carbanolamine intermediate. Unlike other acid-catalyzed additions to carbonyls, the reaction does not stop here, particularly if we have some way to remove water from this reaction. Instead, the mechanism continues to remove the oxygen completely and form the double bond between nitrogen and carbon. Now this carbanolamine intermediate is protonated. This turns the poor leaving group of the hydroxide ion into a good leaving group, water. In the next step, the water leaves to form the carbocation. This carbocation is resonance stabilized. Notice that the other product of this step is water. In the final step of the mechanism, the nitrogen is deprotonated by another molecule of amine to produce the imine product. Remember that the other product of this reaction is water, meaning that this overall process is a dehydration reaction. Removal of water from the reaction mixture helps to drive this reaction equilibrium towards the imine product. In addition to being useful in organic synthesis, imines are common in biochemical mechanisms as well. In particular, we commonly see the iminium ion, which is a protonated form of the imine. The nitrogen, with its positive charge, is a good electron acceptor, making it a useful and intermediate in many biochemical mechanisms. The imine formation, in addition to being a dehydration reaction, is also considered a condensation reaction. A condensation reaction is the joining of two molecules with the loss of a small molecule, like water. Let's look at the overall reaction for imine formation again. This reaction is an equilibrium. Therefore, Le Chatelier's principle applies. We can control the direction that the equilibrium lies by changing the concentrations of the reactions or the products. For example, if we want the equilibrium to lie to the right to favor the formation of the imine, we can either increase the concentration of the amine or decrease the concentration of water by removing water from the reaction mixture as it is formed. If, instead, we would prefer to favor the formation of the ketone or aldehyde, we can either increase the concentration of water or decrease the concentration of amine by removing it from the reaction mixture as it's formed. The formation of the ketone or aldehyde, the reverse reaction of what is drawn, is also called imine hydrolysis. The mechanism for imine hydrolysis is exactly the opposite of the mechanism for imine formation. Remember that the imine is the nitrogen analog of a ketone or an aldehyde. We can add ammonia to a ketone or an aldehyde under acidic conditions to produce an imine, or we can add a primary amine to a ketone or aldehyde. This also produces an imine. This mechanism works with other amine derivatives as well. For example, if we react a ketone or an aldehyde with hydroxylamine, under acidic conditions, we produce an oxime, which is the hydroxylamine analog of an imine. We can also react a ketone or an aldehyde with hydrazine under acidic conditions to produce a compound known as a hydrazone. A hydrazone is the hydrazine derivative of a ketone or an aldehyde. It is important to note that it doesn't matter what is attached to the nitrogen, whether it be protons, another carbon, an oxygen, or a nitrogen. The mechanism for the formation of all of these imines and similar related molecules are the same.